Good afternoon. How are you? Can you hear me?
জি স্যার স্যার কোশ্চেনগুলো কি স্যার কুইজের?
Yes. Can you hear me? First tell me whether you can hear me or not.
জি স্যার।
Okay, great. How are you?
So we are fine, sir. How are you, sir?
I am also fine, and today we shall start a new topic. And before going to that topic, you are asked to answer some questions. So please check your chat box, and you have to reply to my email address. I am giving you the email address.
এটা কি এখনই পাঠাইতে হবে স্যার নাকি ক্লাস শেষে?
Good question. You need to send it now, and to my email address. It would be virtually interactive. So what you need to do, a very brief answer. What is CRISPR-Cas stands for? Who were awarded Nobel Prize in Chemistry 2020 for discovering the CRISPR-Cas genome editing technology? Third question, what is genome editing? Fourth question, what can we do by CRISPR-Cas technology? So just send me a brief email with answers of this question. You have how long time you need to do the
Sir, আবার একটু বলা যাবে স্যার?
Sir, can you repeat the question?
Why?
Sir, repeat the question.
It is available in the chat box. Please go to the chat.
স্যার চ্যাটে স্যার যারা শুরু থেকে ছিল না স্যার তারা মেসেজটা দেখতে পারবে না স্যার।
Really?
জি স্যার, যেমন আমি স্যার ফার্স্ট কোশ্চেনটা দেখতে পাচ্ছি না স্যার, বিকজ আমি ওই সময়টা ছিলাম না, আমি সেকেন্ড কোশ্চেন থেকে দেখতে পারতেছি।
Then it's a problem.
জি স্যার, হয়তো স্যার যারা লেট করে আসবে তারা মেসেজ
স্যার একটা প্রশ্ন দেখা যাচ্ছে
স্যার আমরা ক্লাসের পরে
স্যার
অ্যানসার
আরেকবার রিপিট
দিলে
করলে
হবে?
ভালো হয়।
ক্লাসের পরে আনসার দিলে হবে না, এটা ক্লাসের ভিতরে এখন আনসার দিতে পারলে ভালো।
স্যার তাহলে কোশ্চেনগুলো একটু আরেকবার
ফার্স্ট কোশ্চেনটা সবাই আনসার করো, আমি দেখি।
স্যার কোশ্চেনগুলো স্যার গ্রুপে দিয়ে দেন স্যার।
What CRISPR-Cas stands for? What CRISPR-Cas stands for? You can send me chat box. So what is necessary? Write your registration number and then chat, because all the chat automatically recorded in my computer, so I can even check later on. So what CRISPR-Cas stands for?
স্যার এটা কি স্যার আপনার চ্যাটে সেন্ড করব আপনাকে স্যার?
Yes, write your registration number and then answer instantly.
ইমেইলে দিলে স্যার সমস্যা আছে?
No need mail, you can do it now. What CRISPR-Cas stands for, because it is an abbreviation, so you can elaborate it. CRISPR-Cas, what does it mean? Not your registration number. Some of the
Sir
are sending only registration number. I need registration number and answer. Yes, one student, Anika Anjuma, rightly said.
স্যার
Yes.
স্যার আপনি তো স্যার বলেছিলেন প্রথমে স্যার ইমেইলে পাঠাতে, এখন কি স্যার আমরা চ্যাটে পাঠাবো না স্যার ইমেইলে পাঠাবো?
Chat. Send it through the chat box. What CRISPR-Cas stands for? Yes, people are sending correctly, but what, not only CRISPR, you should elaborate the Cas also. CRISPR, one abbreviation, hyphen, then Cas. What Cas stands for also you should include. Very good. Some of the students are doing good job. What CRISPR-Cas stands for? Yes. Some students did very good job. Yes. Great. You have 30 seconds more time to complete this question. To answer this question, yes. Great.
স্যার বৃষ্টি হয় কিছু স্যার প্রশ্ন শোনা যায় না
I am speaking very loudly. What CRISPR-Cas stands for? You have only 20-second time left to answer the question through the chat box. And you must write your registration number.
স্যার আমারটা কি পাওয়া গেছে স্যার? আমি তো প্রাইভেটলি সেন্ড করলাম স্যার।
Yes, I received all of them. No, please do not answer other questions now. Only what CRISPR-Cas stands for. Others I will ask you later on. Okay. Time is over. I received lots of answers. I will record and check it. But can anyone tell me what CRISPR-Cas stands for? Just unmute yourself and tell me.
Clustered regularly interspaced short palindromic repeats.
It is CRISPR. And then what Cas stand for? CAS. I asked question what CRISPR-Cas stands for. So you told the
CRISPR associated.
CRISPR associated. I cannot hear you.
Sir. CRISPR associated protein.
Yes, CRISPR associated protein. Then what CRISPR-Cas stands for? Can you tell me the full abbreviation?
Sir, clustered regularly interspaced short palindromic repeats. Then CRISPR associated proteins.
Excellent. Clustered regularly interspaced short palindromic repeats. CRISPR associated protein. Fantastic. Next question. Who were awarded Nobel Prize in Chemistry 2020 for discovering the CRISPR-Cas genome editing technology? So you can answer now. Just write the name. And first your registration number and then write the name and send me private message, not publicly. I can tell you two lady scientists got the Nobel Prize. So you should not write a single name. Yes. Excellent. Next 30 seconds you have to complete the answer of this question. Okay, time is over. Can anyone tell me who received the Nobel Prize in Chemistry 2020 for discovering the CRISPR-Cas technology?
Sir Emmanuel Carpenter and Sir Jennifer A. Doudna.
Emmanuel Charpentier and Jennifer Doudna. Emmanuel Charpentier, do you know from which country?
UK, sir.
Emmanuel Charpentier from France and Jennifer Doudna,
France.
US.
Emmanuel from France.
Yes, and Jennifer from?
America, sir. America.
Yes, USA, United States of America, exactly. Next question, what is genome editing? Please write your registration number and then answer the question. Only one sentence, so you need 1 minute or 1.5 minutes. Excellent, some of the students are very fast. Yes, next 30 seconds you have to complete it. Excellent. Most of the students are answering correctly. Excellent job. And your last question, what can we do by CRISPR-Cas technology? We can do lots of things. Just answer one. What can we do by CRISPR-Cas technology? We can do lots of things. Just answer one that can be done by CRISPR-Cas technology. Can anyone tell me what can we do by CRISPR-Cas technology? Excellent. Okay, time is over. Can anyone tell me what can we do by CRISPR-Cas technology?
So we can use it for genome editing purpose. So DNA sequence to alter করার জন্য অথবা modify gene
Yes,
function.
we can alter the DNA sequence of any organism. That means we can rewrite the life. People thought that what is blotted cannot be blotted. That means what is written in DNA, you cannot change it. But CRISPR-Cas technology helps us to rewrite the life. Life means the nucleic acid DNA, you can rewrite, you can change, you can make a change in the DNA. And what you can do, you can design an organism. For example, design a baby, design a plant, and design an organism. And CRISPR-Cas has been used in almost all organisms, including human. Okay, so it is a revolutionary technology for bioediting. Just like textbook or Microsoft Word or Photoshop, you can edit in the DNA. Editing in the DNA means you are changing the traits of an organism. Traits means character, feature, special feature. For example, you know, European people have blonde hairs, and Asian usually black hairs. So if you change the DNA sequence responsible for the blonde hair, you can make European with black hairs, like that. So it is an amazingly powerful technology which was discovered first in 2012 by two ladies, Emmanuelle Charpentier and Jennifer Doudna. And this technology received you know, recognition in 2020. That means this year, and they won the Nobel Prize in chemistry. And there is a CRISPR Fest is going to be held in Bangladesh. CRISPR Fest, and you may find in the Facebook. If you browse it, CRISPR Fest, you can participate on it. Many scientists, you know, big scientists, they will participate. And on 26th, we invited a giant scientist, Sufyan Kamaun, to talk about disease resistance in plant by editing the genome using CRISPR-Cas. And Sufyan Kamaun from UK, he's a Royal Society fellow, very powerful scientist, and he is our collaborator who first edited the genome of plant by using CRISPR-Cas. So you may enjoy his talk, possibly a Facebook Live or like that, and I shall be also there. So this is all about CRISPR, and I hope you enjoy it. Those who missed to answer the questions, 5 questions correctly through the you know, chat box, I shall you know, save the chat box all messages, but you have an alternate opportunity to answer all 5 questions through email. So this is a second option. My target is to see how efficiently you learn the new topic. And CRISPR, I found everywhere, young people love CRISPR very much because it is really a crazy technology to rewrite the life. You can change any character of an organism by using CRISPR. So we shall start another Nobel-winning topic today called autophagy. And this topic also received Nobel Prize in 2019. possibly 16. Yes, autophagy. Do anyone heard about autophagy? What is it? Can anyone tell me what does it mean? Autophagy. Any idea?
নিজেকে নিজের খাওয়া
O2 means
cell breakdown and renew of cell, new cell.
excellent. O2 means self. Phagy means, phase. It comes from phase. That means eating. Self-eating. Self-eating means in the cell. In every cell, there are lots of organelles. As a biologist, you know what are the major organelles in the cell. For example, nucleus, mitochondria, cytoplasm, endoplasmic reticulum, lots of Golgi apparatus, lots of plastid, lots of organelles are present inside the cell. Autophagy, this is a process actually. This process always active in the cell to clean the waste generated by the cellular activity. Metabolic activities, so it is recycling process, recycling of the waste in the cell. Very fantastic, discovery, and at cell level, how recycling process is going on. Scientists discovered it, and they got the Nobel Prize. You may ask a question, what is the meaning? So we shall discuss what is the meaning of autophagy, how it is related to our life and life of other organisms. The scientist who got the Nobel Prize, his name is Yoshinori Ohsumi. Yoshinori Ohsumi received Nobel Prize in 2016 for discovering the process of autophagy in the cell. And he is basically a microbiologist in Japan. So he received the Nobel Prize. And if you browse the internet, you can find all the story. So what is autophagy? The word autophagy originates from Greek words auto, meaning self, and phagein, meaning to eat. Self-eating. So it is self-eating process which degrades and recycles cellular components. Cellular components, you may ask, is it a cell organelle can be eaten by the autophagic process? Yes, if it is useless, then it is an waste. For example, you have shoes in your house. Some of the shoes you are not using and they are useless. So not keeping in your house, better to recycle it. So autophagy.
Sorry.
Yes.
Sir, sorry for interruption. Sir, হচ্ছে আমি স্যার মানে পাঠাবো হচ্ছে মেইলে পাঠাইতে যায় আমার হচ্ছে ডিসকানেক্ট হয়ে গিয়েছিল আমার মানে বোনের আইডি থেকে স্যার আপনার মেইলে একটা হচ্ছে মেইল পাঠাইছে স্যার।
All right, no problem. Elaborately, it is a process whereby the eukaryotic cell can recycle part of its own content by sequestering a portion of the cytoplasm in a double membrane vesicle that is delivered to the lysosome to degradation. This is very nicely and very easily I told the story. What it happens, in the cell, there are lots of unused materials. For example, if you take antibiotics for, treating any disease, in that case, where, antibiotic goes to the cell and it kill the, microorganism, but pathogenic microorganism. Then what happens to the antibiotic? That antibiotic is, recycled or, it is discarded from the cell by the autophagic process. How did it happen? all the waste in the cell is wrapped up by double membrane vesicle-like structure is formed to wrap up all the unused, useless traces or waste present in the cell, and then they use different enzymes to make to recycle them. To recycle means, for example, a protein. A protein can be recycled, make a new protein. So break down the protein and make all the amino acids, because amino acids are needed for synthesis of new protein. So you can recycle it. For example, plastic, glass, and metals, we can recycle in our practical life. So similarly, useful biochemicals and organelles which contains valuable raw materials of preparing new biomolecules in the cell, they are recycled in the autophagic process. But there are some bad things. For example, COVID-19 virus, if entered into the cell, it is not possible to recycle, because cell cannot use the RNA of COVID-19 to make something important. In that case, what would happen? In the autophagic process, the RNA of COVID-19 and the protein code, they are break down and destroy them and then delivered to the lysosome. Lysosome is the main dumping place of the cell waste for the digestion. Lysosome digest all the unusable or recyclable waste for digestion. Digestion means make small molecules like carbon dioxide or water and throw out from the cell, coming out from the cell, and they are excreted by urine or stools or sweating or other gaseous form. So removed from the cell or organisms. So autophagy is really very interesting. If I ask you the process of autophagy, do you have the process of autophagy in your cells? The answer should be yes. And the autophagic process are always going on in all eukaryotic organisms. Your cells, also in my cells. But in older people, autophagic process are slower. In younger people, for example, your autophagic process is more efficient than mine. So you can imagine that autophagic process, recycling process, is very important for vitality and stamina of the life. So the history of autophagy starts from the discovery of Christian de Duve, who was a Belgian scientist who was interested in the action of insulin and studied the intracellular localization of glucose 6-phosphatase enzyme using cell fractionation method developed by Albert Claude. This is another guy. These two guys, very famous cell biologists. The Duve observed a new specialized cellular components called an organelle containing enzymes that digest proteins, carbohydrates, and lipids called lysosome. That means it is clear that Christian de Duve first discovered the lysosome. Lysosome can digest protein, carbohydrate, and lipid. Useless protein, useless carbohydrate, useless lipid. That means they are the waste in the cell. They are dumped into the lysosome. And lysosome functions as a workstation for degradation of cellular constituents. So you understood that lysosome was discovered much earlier than the autophagic process. So lysosome was discovered by Christian de Duve. And Christian de Duve, obviously, and Albert Claude, they got the Nobel Prize for their discovery. And de Duve coined the term autophagy in 1963. Coined the term means he first proposed the term autophagy in 1963. At that time, what is autophagy, why it is important, what is its function, which genes are responsible for this process, nothing was known. And extensively discussed the concept in a review article published a few years later. That means de Duve first, proposed this term and discussed a lot. But at that time, he couldn't figure out what is autophagic process. Otherwise, he could get another Nobel Prize. Christian de Duve was awarded Nobel Prize in Physiology or Medicine in 1974 for the discovery of the lysosome with Albert Claude. This means these two guys got the Nobel Prize in 1974 due to their discovery of lysosome. Lysosome is the main dumping station for in the cell for digestion of the waste materials. So after coining the term autophagy, a lysosome-mediated intracellular degradation pathway was discovered in early 50s, and cell biologists exploring the mammalian cell with then newly developed high-resolution transmission electron microscopic technique were the first to observe in cell lysosome-like membrane-bound structure that contain organelle like mitochondria. Very interesting. You see, by using transmission electron microscopy, because normal microscopy cannot see the autophagic body, that is the vesicles, because they are too small. They use transmission electron microscopy. You can do the section of the cell at nanomolar level, nanometer level. You can see the organelle, and they found some organelle, lysosome-like membrane-bound organelle present in the cell, but they couldn't figure out clearly. Later, those structures are called cytolysosomes and next autophagosomes. Autophagosomes recently discovered a term. Previously, people thought cytolysosome. So they are not actually lysosome, but lysosome-like structure called autophagosome. In 1963, the term autophagy was coined by de Duve. Already I told you. After that time, it was impossible to imagine how broad and significant the influence of autophagy would turn out to be in future. And autophagy is today recognized as a major quality control pathway and metabolic regulation system in the cell. And in essence, autophagy proceeds when a selected part of the cytosol is engulfed by a double membrane, forming the so-called autophagosome, which later fused with the lysosome. That means autophagosomes are vesicle-like structure, double membranous structure like lysosome, and they are separate, structure, and they are finally dumped into the lysosome. But autophagosome can recycle some of the unused cellular materials, and it is a digestive organelle of the cell where a number of catabolic enzymes facilitate the breakdown of cargo, enabling recycling of the generated metabolites. So yeast was used to discover the autophagic process by Yoshinori Ohsumi, because yeast cells, yeast is fungus, and yeast is has been used as biotechnology since ancient time, because yeast is used in making the bread. So yeast cells are relatively easy to study. Consequently, they are often used as a model for human cell because it is eukaryotic, and yeast is a model eukaryotic organism and easy to handle. Life cycle is quick. particularly useful for the identification of genes that are important in complex cellular pathway. So you shouldn't use the yeast to discover the autophagic process, but autophagic process exists in all eukaryotic organisms. What were unknown in 1990s? Even in 1990s, almost 30 years after de Duve coined the term autophagy, the process remained a biological enigma, that is an illusion, unknown. Molecular markers were not available at that time, and components of the autophagy machinery were elusive, not known. But many fundamental questions remained unanswered. How was autophagy process initiated? This was the first question, research question. How was the autophagosome, that is double membrane structure, vesicle-like structure, is formed? And how important was autophagy for cellular and organismal survival? How autophagy is important for the cell survival as well as survival of the organism. Did autophagy have any role in human disease? These four questions, fundamental questions, are unanswered in early 90s, even 30 years later of the discovery, the coining of the term autophagy. But after 1990s, scientists discovered, answered all these four questions. So usually answered all these four questions. Although biochemical and morphological phenomena of autophagy fascinated the cell biologist, nothing is known about underlying molecular mechanism. You may ask me question, what is underlying molecular mechanism? That means the metabolic pathway. That means the gene involved there, which gene regulate and express during autophagic process. Several genes are involved there. Several metabolic pathways are involved to recycle different kinds of, cell waste. So all these molecular mechanisms are unknown. Oshumi approached this problem by studying autophagy in yeast, in which the lysosome-like vacuoles are endpoints of the autophagy pathway. In the 1980s, discovered by cell biologists working in the model system Saccharomyces cerevisiae. Saccharomyces cerevisiae is the model yeast, scientific name of model yeast, and progress was being made in understanding transport to and from the vacuole of the cell. And Oshumi identified autophagic bodies in nitrogen-deprived yeast Saccharomyces cerevisiae. That means yeast which received less nitrogen. Nitrogen is essential for all cell, all organisms. So he deprived nitrogen, offered less nitrogen to the Saccharomyces cerevisiae, and was the first to report the nutrient deficiency induced autophagic degradation in yeast. Very interesting. You see, when nitrogen was not provided enough, in that case, autophagic process was accelerated to get the nitrogen from waste materials, because some proteins, some other, cellular materials that contain nitrogen. So autophagic process accelerated and degraded and release the nitrogenous compound required for the metabolism. So when we do fasting during holy month of Ramadan, our autophagic process is accelerated, and then waste materials in the cell are cleaned, because daytime we cannot eat protein, milk, and other proteinous substances. So we are starved for nitrogenous materials. You may ask question whether fasting is good for health. Yes, fasting is good for health. Your autophagic process, recycling process is accelerated. A key point in this approach was the use of vacuolar protease inhibitors. Protease, involved in protein metabolism. He used protease inhibitor to allow for accumulation of intermediate autophagic bodies, the inner membrane bound content of the double membrane autophagosome inside the vacuole. So Oshumi's lab characterized these structures by transmission electron microscopy and determined which proteases were required for the degradation. This seminal observation established, seminal means the fundamental first observation, established the system and facilitated the first genetic screen for yeast autophagy mutants that was carried out in proteinase deficient yeast cell to enable detection of the short-lived autophagic bodies. The mutant isolated were originally called APG. You may ask me question, what is mutant? Mutant means which is not able to process the accelerate the process of autophagy. So somehow Oshumi's lab caused the damage in the DNA and made some yeast which has no ability to autophagy. That means he called them APG mutant. That means that mutant is defective. That means some genes are somehow damaged which are responsible for autophagic process, and that helped APG11 mutants are identified by their defect in accumulation of autophagic bodies, and Oshumi's lab observed that they lost viability much faster than under conditions of nutrient deprivation. Very interesting. You can understand, as they are mutant, they are unable to do autophagic process. When you do not offer the nutrient, they cannot digest the cell waste and offer supply the nitrogen. This is why those cells lost their viability. They were died. That means if autophagic process somehow in our cell becomes dysfunctional, we cannot survive. So those mutants confirm that which genes are involved in autophagic process. With this groundbreaking work using mutants, the Oshumi lab identified the first autophagy gene and showed that there are at least 15 needed in yeast. 15 needed means 15 genes are involved in the autophagic process. So not a single gene, because those that APG11 mutant lacked 15 genes. This is why unable to do autophagy compared to the normal yeast. So they published their findings in a journal of cell biology. Autophagy in yeast demonstrated with proteinase deficient mutants and conditions for its induction. It was published in Journal of Cell Biology in 2nd October 1992. And this publication brought the Nobel Prize in Physiology and Medicine. So this is the concept of autophagy. Later lecture, I shall tell you the mechanism of autophagy and how autophagy is important for tackling the diseases, even cancer, Alzheimer, and many other diseases, and how autophagy is related to the vitality or the sound health of any organism, and how autophagic mechanism and understanding can be utilized for designing new drug to keep us healthy. So I shall discuss the other points later on, because I think the whole story would be too much for you for today. So we shall have some time for asking the question. So you can ask some question now. It is your, floor is now yours.
So can you please tell me what will be the result if autophagy process is dysfunctional in our body?
Autophagic process obviously functional in our body. In our body means autophagic process, you must understand, it is functional at cell level. DNA is functional at cell level. Cell is the unit of organism. Like a brick is a unit of a big building or like that. So autophagic process is functional in our cell, and our cell use autophagic process to recycle the waste. And the process, if the autophagic process is faster, efficient, it means you are younger, you are smarter, you are strong. But if autophagic process is weaker, you will become older, you will become weak, or like that. So autophagic process is happening at the cellular level, but its impact is at the organismal level. Any more question? Terin, okay? Yes, Tahmina, you want to ask a question?
স্যার টিউমার সেল যে গ্রোথ হয় অনেকের হচ্ছে আমি দেখেছি টিউমার সেল আমার এক রিলেটিভেরই স্যার হাতে হচ্ছে টিউমার সেল হয়েছিল, তো সে একবার অপারেশন করেছে কিন্তু তারপরেও স্যার তার মানে ওই গ্রোথটা স্যার কমতেছে না, হয়তো মানে কিছুদিন পর আবার হচ্ছে। এটা কি কারণে হতে পারে? কোনো মিউটেশনের কারণে অথবা কোনো আদার্স প্রবলেম?
Thank you very much. The cancer is associated with undesirable mutation in the DNA, and it may happen through genetically, or it may happen different due to the exposure of different carcinogenic materials. It may happen through radiation and many other reasons, even error in coping system of the DNA. So tumor is a symptom of cancer. That means if mutation taken place in certain location of the DNA, then certain tumor or cancer is the may result some cancer or which is expressed by the tumor. So you may cut the tumor, but you cannot, if you do not repair it at the DNA level, it may reoccur. That means recurrence. But some of the tumor, for example, breast cancer or breast tumor, or tumor in some other local tumor, if you remove it, in most cases, for example, thyroid, tumor in the thyroid gland or like that, you can remove them by surgery, and in most cases they are not recur. But as DNA level, mutation is remained, so there is a chance of reappearance or recurrence, like your the relatives. Tumor at initial stage, if you remove, no problem. But if tumor cells, they do not abide by the process of apoptosis, another process, programmed cell death. This is why they only know the proliferation and uncontrolled disobedient cells. If they migrate to the vascular system, that is blood circulatory system, then it is called metastasis, and it is called last stage of the cancer. And at that stage, we cannot treat the patient. Usually as they grow in different organs and different system, and they disrupt the metabolic processes and cell division and others, and quickly kill the patient. So a tumor we see is a symptom, but the real switch of developing a tumor is the mutation in the DNA. Nice question. Next question.
So, sir, is this only applicable for the eukaryotes, and what's about the prokaryotes one?
You mean the autophagy? Autophagy is found in the eukaryote, because eukaryote contains a cell contains karyon. That means nucleus is well structured. That means DNA is enveloped with the double membrane structures, and many other organelles, they are membrane bound structures. So eukaryotes are advanced, and eukaryote cells are very complex. But if you see prokaryote like bacteria or archaea, very simple, and they have no need, how to say, single cell, single organism. So they have no need, possibly, the delicate autophagic process. Bacteria from 1 to 2, they can immediately multiply. Even in the petri dish, you can make billions of copies of a bacteria in culture medium. But eukaryotic organisms like human or complex animals, their cellular or plant structure is complex, and till now autophagic process found very effective in the eukaryotic system.
Sir, I have a supplementary one, sir.
Yes.
Sir, as far as I know, sir, plants don't have the lysosomes. So, sir, what's that in this case?
Yes, autophagic process, I shall show you later on. Autophagic process are there. And plant has also mechanism to recycle the materials, and good through the autophagic process. I shall discuss about your question, how plant handles other materials, and how plant has the excretory mechanism. Plant has not excretory mechanism like we have. Plant is smarter than us, because they can get the energy from the sunlight to make their energy containing chemical substances in their cell through the photosynthesis. And what we are doing, we are eating plants, animals, and all the energy sources. The main origin is the sunlight, and main factory of converting sunlight into chemical energy is the plant. So plant has very sophisticated and smart system for handling their own waste. This is why you can see a plant can survive, some of the plant can survive several 1,000 years, but you cannot find other eukaryotic organism can survive so long time. But obviously there are some plants, they are short duration, and plant, very easy to get do tissue culture of the plant. From a single cell you can have a full plant, but from a single cell of human you cannot find get a human baby. We have not yet been discovered such a technology, although stem cell technology have been discovered several years ago. So plant has smarter system. I shall discuss in later section of the lecture more detail. Very nice question.
Okay,
Thank you.
sir.
Any more question?
Sir, I have a question, sir.
Yes, please.
Sir, you said autophagic process is positive in our body.
Yes.
Have any factors that speed up or boost up autophagic pathway in our body?
Yes, autophagic process is good for our body. But autophagic process in the cancer cell is bad for us. If autophagic process in the cancer cell is more powerful than autophagic process in our healthy cell, then it would be terrible. So in case of cancer cell, scientists are trying to discover the autophagic processes in cancer cell and the healthy cell. Are there any differences to find the molecular switches so that scientists can use a drug to stop the autophagic process in the cancer cell to treat the cancer? Then cancer cell cannot divide and they can become the old. In our body, autophagic process, whether can be accelerated or not, drug is not yet, a single drug is not yet discovered, but there are some, molecular switches have been identified, and if it continues to the development of powerful medicine in future, maybe we can extend the longevity of our youngness by accelerating the autophagic process in our body. But what you can do exercises, you can do physical work to accelerate the autophagic process. By doing physical exercises, you can also maintain a sound, how to say, or balanced diet for accelerating the autophagic process. For example, fasting is also accelerate the autophagic process. This is why it is good for health. Do you know, in the night, in Western people, the breakfast, morning food, they called breakfast. Do you know why? Because they eat, for example, German. I have been in Germany for 2 years. German people takes their dinner at 6 to 7 o'clock in the evening, and whole night, and they take very less in the dinner, very less, just like a small bread or like that, and whole night they are doing fasting, and in the morning they do breakfast. That means breaking the fast, like our iftar. So it is a practice to accelerate the autophagic process. But if you keep you hungry irregularly for longer time, then you may be affected by acidity and some other problem. Fasting, there is a routine, there is a certain period of time. Do you know the difference between hatahati and hata? Walking. For example, a cook in the house, in the kitchen, walk a lot for cooking and moving, and a maid servant in the house do lots of work and walk a lot. But it is not the real exercise. For real exercise, you have to walk in a certain speed for a certain period of time, and routinely you have to do it, so that sweating and your all organs and cellular level metabolic activity is accelerated, autophagy is accelerated. Then it is, I call, hata. Otherwise, hatahati is not useful, but hata is really useful for your health. Any question? Very nice question, Abdul Rahman.
Sir,
Abdul Rahim.
sir, if the elderly people start to fast twice a week, will their autophagy accelerate?
If the
elderly people whose autophagy speed has been reduced, if they fast twice a week, will that accelerate their autophagy process?
It's a good question. Older people, older people aging, to say you sadly, aging is a natural process, and aging is associated with the function of lots of genes in our body. So older people, you can slow down the aging process. you can slow down the aging process by two ways. One is changing your lifestyle, and another is changing your food habit. So if you can maintain a standard lifestyle, that means do regular exercise and eat routinely and maintain required energy and take diversified food to satisfy all the requirements of micronutrient, vitamins and other minerals. And then what you can do, you can keep you younger for longer time. Aged people, if they do some exercise, if they do fasting, you mentioned fasting, at certain stage, fasting obviously help them. But when you become older, the mitochondrial activity also, and number of mitochondria per cell also decrease. So in that case, not only fasting, if you combine with fasting, lifestyle, and also food habit, then you can accelerate your autophagic process. And another point is to accelerate the mitochondrial activity. For example, your friend who is doing regular exercise and you are not doing regular exercise, even in the same age, if you check the number of mitochondria in your cell, number of mitochondria in your friend's cell, you can find different. Mitochondria number as well as activity increase when you do regular exercise. It is very, very important. And if you can start from your current age, I can guarantee you that you can avoid lots of diseases. You can keep you younger rather than using synthetic cosmetics. If you do regular exercise and maintain a good lifestyle, including food, then you can accelerate your youngness, and you can accelerate your autophagy, you can accelerate the function of your mitochondria, and you can accelerate your vitality and stamina. And it is known, but most of the people, it is known discovery, but most of the people, they are lazy and they do not care about it, especially at your age. For me, at my age, after 50, when I realize it, no meaning. But if you realize it, you can impact in your life robustly. Thank you so much. Very nice question. Any more question? Very exciting question. Rumi Khatun, do you hear me? Musammat Rumi Khatun? Please unmute yourself and tell me whether you hear me or not. Can you hear me? I cannot hear you.
Sir.
Yes, fine. What is autophagy?
Sir.
Yes, walaikum salam. What is autophagy? Rumi Khatun, what do you understand by the term autophagy?
Sir.
Yes, tell us. Whatever you understand.
Autophagy meaning?
You can speak in Bangla also, no problem.
Destroying of cell.
Destroying of cell. Is it correct? Destroying of cell.
Meaning to eat.
Meaning to eat. That means destroying, not destroying. Recycling and destroying of the waste, cell waste, waste product, trash, which are undesirable materials in the cell. So autophagy is a process.
Recycle the cellular components.
Yes, even cellular components. For example, a mitochondria, dead mitochondria, or a dead chloroplast. Autophagic process can engulf it and recycle it and dump it into the lysosome. Very good. So I can ask anyone question if you do not ask me question. So it is better and safe for you to ask me question.
Sir, I have a question, sir.
Yes, please.
Sir, does exercise speed up autophagy, sir?
Yes. Straightforward answer is, if you do exercise, you can accelerate the process of autophagy. If you do exercise, you can accelerate the process of mitochondrial respiration. And obviously, remember that do exercise in a good environment where you can get lots of oxygen, and not in the polluted environment, not in a closed chamber or room. Better to do exercise in the park or in the open air or a place where good aeration is available. Very nice question. So from now, please do some exercise. And those who are not, doing the fasting during Ramadan, you must understood that science behind it, science support the fasting, and you would not be killed by, taking the fasting during Ramadan. Rather, you can enhance the recycling process of your cell and become more, how to say, you will have more vitality after the Ramadan. And Ramadan is obviously you religiously you also have some benefit. Anyway, thank you so much. If you have no question, I should stop here and think about CRISPR Fest and attending the all amazing progress in CRISPR, because you can find lots of stuffs in YouTube, and CRISPR would revolutionize in biology, in our life as well as in agriculture. So I am thinking, how can I engage you and offer you an assignment related to the CRISPR-Cas technology. So maybe after the autophagic lecture, I shall offer you lots of topics for doing assignment on CRISPR, the world latest cutting-edge technology. So you are lucky, you are getting the science in this class, which is groundbreaking for the human civilization, which is groundbreaking for the whole world, groundbreaking for promoting the economy as well as human power in this earth. And obviously, if you get the message from the essence from the lecture, you may also utilize lots of science in your life to make your life more, happy, or more prosperous. Thank you so much.
Sir.
Yes.
Sir, do we have to email the question, sir?
Yes. What you do, I shall check your chat messages. If you think that you made a mistake and not efficiently answered in the chat, I shall also compare your email message. If email message is better than the chat one, I shall give you a good marking over there. So email message is an alternative, not obligatory. So it depends on you. Thank you so much.
মেইল অ্যাড্রেসটা কি আরেকবার দেওয়া যাবে? স্যার আমি স্ক্রিপ্ট করছিলাম, ডিসকানেক্ট হয়ে গেছিলাম। মেইল অ্যাড্রেসটা স্যার।
Email address again. Just a moment. I am giving it through your chat box. So please write it. Oh, it should be everyone. Just a moment. It is also everyone. If I want to do everyone, not private. Yes, I am giving an email address to everyone. Please note it, and later I will not give you email address anymore. You should use my official email address to send your answer. Thank you so much. No question, and wish you a happy day, and obviously stay safe. Thank you all.